transcribe RNAs that are never translated into proteins. Things like the ribosomal RNA, the RNA that becomes part of the ribosome, or the transfer RNA, the RNA that becomes part of the tRNAs, are made from genes that are transcribed by RNA polymerase 1 and RNA polymerase 3. The RNA polymerase 2 is the one that transcribes the genes into messenger RNAs for proteins. So just like we saw in prokaryotes, upstream of codon number 1 is a promoter sequence, which is where the RNA polymerase is going to lock onto the template strand. And just like we saw in prokaryotes, there are some sequences that you find in promoter sequences in eukaryotes. But one of the distinctions is that there's a little bit more variability in the promoter sequences of eukaryotes. In other words, there's not just one particular sequence that you always find in the promoter in eukaryotic genes. There are some fairly common sequences. There's one called the TATA box, which is kind of a funny name. TATA, it's named after the sequence that you find there. And you find this TATA box in a lot of eukaryotic promoters, but not in all eukaryotic promoters. And I don't have it on this diagram, but there's another one called the CAT box. It's also fairly common in eukaryotic promoters, but you don't find it all the time. And there's a whole bunch more that I don't know, that I'm not an expert in. But anyway, the point is there's a lot more variability in the promoter sequences in eukaryotes than prokaryotes. But what I think one of the weirdest things is the RNA polymerase enzyme doesn't bind to those sequences. The RNA polymerase enzyme in eukaryotes does not have a binding site for the TATA box or the CAT box or any other promoter. In and of itself, it cannot latch itself onto the template strand. It just has no binding site for any promoter sequences, which sort of begs the question, well, how does the RNA polymerase 2 get attached to the template strand if it doesn't bind to any promoter sequences? Well, there are some special proteins in eukaryotic cells called transcription factors. And it's these transcription factor proteins that bind to the promoter sequences like the TATA box. And then once the transcription factors have bound onto the promoter, then those transcription factors essentially grab RNA polymerase 2 and attach it to the template strand. So here's RNA polymerase 2. It just doesn't have any binding site for the promoter sequence, and so it just can't attach to it. Here come some of these transcription factors, which do have binding sites for TATA boxes and CAT boxes and things like that in the promoter. And once they are attached, then they grab RNA polymerase 2 and click it onto the template strand. Click. And now RNA polymerase 2 can go about transcribing the gene. And there you have some 5' untranslated region. There's the first codon, AUG. There's the second codon. Onward, onward, onward. Now, you might wonder, well, why is it that RNA polymerase 2 doesn't bind right to the promoter sequence? Why do eukaryotes have to have these transcription factors to help it attach? Essentially, it comes down to this, that the regulation of transcription is a lot more complex in eukaryotes than prokaryotes. Our ability to turn certain genes on, turn other genes off, is more complex and more nuanced than in simple little prokaryotes. And so having a whole bunch of transcription factors that are involved in the process of transcription allows for finer tuning of expression than we find in prokaryotes. Oh yeah, and one final concept, how does transcription end? We have to have some way of ending that messenger RNA. What we're going to see here is that it's not the same as in prokaryotes. All right, so there's the stop codon right there. The RNA polymerase 2 transcribes past the stop codon. so. And it just keeps transcribing right on past that stop codon. Eventually it transcribes a certain sequence called the polyadenylation signal. AAA is the polyadenylation signal. And it actually transcribes past the polyadenylation signal too. But there's an enzyme in the nucleus. Remember, all this is taking place in the nucleus. There's an enzyme in the nucleus that will then find that signal in the messenger RNA and snip the messenger RNA just downstream of that sequence. Polyadenylation signal. And here comes that enzyme I was telling you about. It's like a pair of scissors. It finds the polyadenylation signal and then just downstream of there snips the messenger RNA. And so that's what gives you the end of the messenger RNA. It's always snipped just slightly downstream of the poly-A signal. Now, what happened to the RNA polymerase II that was just chugging along further and further off in the 3' direction? I don't know. Say goodbye to it. Eventually, it does detach itself somehow from the template strand, but how it eventually falls off is not exactly clear. But the point here is, this does give us an M to the messenger RNA right there. All right, that's a good stopping point, and we'll do the quiz now. So put your stuff away, please.